Joining us today is Lindsey Klein. She works with UC Cooperative Extension in my program, Small and Organic Farms program, as the community education specialist. Lindsey grew up in a small town in northern Vermont, surrounded by small dairy and diversified vegetable farms. This early exposure to agriculture helped nurture an interest in community food systems. A few years after college, she moved to California to pursue a master's in international agricultural development from UC Davis. While at UC Davis, she studied how higher levels of soil organic carbon can impact nitrous oxide emissions in agricultural soils, something that well positioned her to explore soil health. While in grad school, she also worked for a local winery in Davis, where she got to partake in everything from irrigation system maintenance to blending and bottling wines and working in the tasting room. In her current position, she has spent the last 2 years focused on exploring the use of soil health tests as meaningful tools for local farmers. So I'm looking forward to hearing what she has learned through this project, and I'll pass it over to you, Lindsey.
Great. Thank you, Margaret. Let me get myself set up here. Okay. Yeah, so thank you all for joining me today and eating some lunch while we talk about soil, which is never a bad way to spend midday on a Tuesday. Um, there we go. So today I will spend the bulk of my time kind of exploring some of these soil health tests a little bit more in depth, but I will start off just kind of with a quick overview of soil health testing, kind of why we're here and give that context. And then at the end, I'll spend some time thinking about how we can actually use these soil health tests to answer questions that we might have. And then we'll have some time at the end for a Q&A. When we're talking about soil health, you'll often hear us talk about these four principles of soil health. And we're trying to both feed and protect our soil. And in order to feed our soil, we talk about maximizing continuous living roots in the soil. We want to maximize our biodiversity, and that's all providing really great food for our plants and soil life. On the other side, we also want to simultaneously protect our soil. And so that involves minimizing our disturbance and maximizing our soil cover. And so when we want to try and figure out how we're doing as we work on all of these things, we can't just turn to one great silver bullet sort of test in order to do that. Instead, we investigate our soil's chemical, physical, and biological health, and we use indicators of these things to kind of indirectly get a picture of what our overall soil health looks like. And so to measure these three different groups, we have a variety of indicator tests that fall into each. Some of these you'll be more familiar with than others. The ones you're probably most familiar with are these chemical tests. And these are the ones that we typically find in our standard soil fertility tests. These are things like our macro and micronutrients, cation exchange capacity, pH, electrical conductivity. We then have these physical indicators. These are sometimes less frequently measured, but they're really important for giving us an idea of what our soil structure looks like, its integrity, and things like that, all of which then influence our soil's ability to hold and move water throughout the soil profile. Of these tests, the ones you're probably most familiar with is your soil texture. This is one that you will sometimes get in your standard fertility test as well, even though it's a physical test. And texture is a really important thing to know when it comes to soil health testing because it provides important contextual clues about how we interpret our test results or what we can expect from certain soil health practices. And so even though we can't change our soil texture, it's an inherent soil property, it doesn't mean that it's not important and not a good thing to know. But some of these other tests that we'll look at are things like aggregate stability and our soil's available water holding capacity. And then the group of tests that we talk about more regularly are our biological tests. And when we're feeding and protecting the soil, we want to know how that's impacting the life. And so we want to know who's down there and what they're eating and how active they are. And so we use these tests like organic matter, microbial biomass, microbial diversity, respiration, which we also call carbon mineralization. to get an idea of what's going on down beneath our feet. And the tests that I have here are not the be all end all. You've probably heard or used some other ones as well. There are many different tests that are out there, and trying to kind of figure out what the best one to use is kind of the conversation du jour. That's what we talk about a lot is what tests you should be using. And again, is kind of why we're here. Some organizations, like the Soil Health Institute, have taken a stab at making that determination. And these are three that you may have heard them talk about pretty regularly. And they suggest that you measure aggregate stability, organic matter, and carbon mineralization. And so you'll see that these tests are falling in our physical test category and our biological test category. We don't really see anything here in the chemical one. And that doesn't mean that they're ignoring it or that it's not important. It's just that these organic matter tests and the carbon mineralization tests are also indirectly measuring some of these chemical things in our nutrient availability, in our nutrient cycling. And so they're trying to encompass that all in one. And so because we do hear from the Soil Health Institute about these tests quite a bit, and it might be something you've come across, I did just want to make sure we point that out. That's not to say that like these chemical fertility tests aren't important. I think they're actually really, really important and something that we can sometimes overlook. But these are really important tests that we use as tools when making our management decisions on the farm. And then also when we're thinking about soil health, we're thinking about like building an ecosystem that's sustaining our plants, animals, humans, the world around us. And we can't do that if our soil fertility is off. We can't sustain these plants and animals. And so this entire system is dependent on having overall soil health and not just focusing on one or two. And so we can kind of think about it as like a barrel full of water. Some of you might be familiar with this diagram, but basically what it's saying is that our yield, or we can think about how good our soil health is not dictated by the total resources available. It's not dictated by I'm sorry, it's not dictated by like how good all this is overall. Instead, it's managed by what is our most scarce resource or our most limiting factor. And so if we think about this in soil health, our soil health isn't just dictated by how good our aggregate stability is or how good our soil biological health is alone. Instead, we need to think about the holistic thing because if we forget about pH or we forget about our nutrients, we're not going to be successful. And so that's why I think it's important to kind of bring this all together and think about all three of these different groups all together rather than ignoring one or two in favor of another. And so a lot of the labs that are out there will try and capture this in their different test packages. This is like a pretty kind of overwhelming list in some ways of just some of the soil health labs that are out there and soil health packages that are being offered. And you'll see that there's not really one, set package. They all have their own variations on this theme. And there's no just like standardized soil health testing package. Each of the tests that are included here might tell you something different. And it can be kind of a lot to, I think, like wade through all of this. But really, like one important thing to do is just like pick a lab and stick with it. And part of that comes down to the fact that even if you're getting a test, say, your aggregate stability test from alluvial soil lab, maybe you want to switch to the soil health lab or something else. They may not be using the same methods to test those things. And so the results that you get back may not actually be all that comparable. And so find one that works best for you. You like their results, you like what's going on there, you like the package, and just stick with that. Another important thing to note with some of these soil labs is that they may give you this soil health score when you get your test results back. Um, and I tend to urge people to be cautious when using these, mostly because when they are developing and calibrating these soil health scores, they're basing them off of soils from the region where that lab is based. So if you have a lab in the Midwest, they're going to be calibrating this score that they're giving you based on data that they're getting from their region. And because soil health is so affected by our inherent climatic properties, it's going to vary. So, what soil health looks like here in Yolo County in Northern California might be different than the Southeast United States, the Northwest. It's going to all look pretty different. And so, rather than look at this score and be like, man, my soil organic matter compared to somebody in the Midwest is telling me I'm doing a bad job. Well, you know, 4% here is going to, be pretty good, whereas in the Midwest, they might be looking for something much higher. And so I would suggest kind of using that as your maybe your own internal kind of scoring guide where you're like, okay, this year I got a 32. Next year I got a 36. The year after I got a 37. And you kind of use that score as your own kind of internal gauge of how your numbers are changing over time, rather than trying to compare yourself to somebody who might be in a different region of the world, or sorry, of the country. So that's not to say it's not helpful, but just kind of a word of caution when it comes to that. And so when we look at these lab offerings, even though, as we saw, there's not just one standard soil health test package, they do tend to kind of fall into some broader categories. We have these organic matter pools and processes, we have nutrition and toxicities, and then we have these kind of physical habitat tests. And I'm going to dive into these categories kind of over the course of my remaining presentation today. So, our soil organic matter, arguably one of the most important, if not the most important, measurements that we take when we're looking at soil health. And you can break it down into some different pools. The largest pool is our total organic matter. This is a pretty large, relatively stable pool. We don't see a lot of, it's long-term changes occur here. We don't see a lot of kind of quick fluctuations in this. The carbon and nitrogen that is included in this pool serves as food for microbes and plants, but it's going to take some time and effort to get that carbon and nitrogen into a form that they can use and take up. And so you can kind of think about this as wheat or milk, where we can't just really go out to a field and eat a stalk of wheat, or we can't necessarily, take milk straight from the cow and drink it. Typically, we have to process the wheat, or we have to, take the milk, pasteurize it, things like that. And so we have to take some time in order to make that available for our uptake. And that's the same way that we can think about the carbon and nitrogen that's in this larger total organic matter pool. And we can measure this carbon and nitrogen by heating it up in an oven to a really high temperature. And we can measure the difference in the weight between our samples afterwards, or we measure how much carbon and nitrogen is given off during that process. From there, we have a smaller pool. This is a more labile, a more available pool of carbon and nitrogen that changes a little bit more quickly. We're thinking kind of in the time frame of by months. And in this pool of carbon, we measure kind of two different things. We look at these pools of carbon. This is what we call permanganate oxidizable carbon. Or we look at ACE protein, and that's autoclave extractable nitrogen protein. And what these are telling us is about the food that's going to be available for our microbes. And so this food here is a little bit more available and easier for our plants and microbes to access. We can kind of think about this as food in our pantry. So think of like your, bag of Orville Redenbacher popcorn or your macaroni and cheese, where it takes a little bit of effort to get it into an edible form, but not a lot. You're not doing as much work to get there as you are with that wheat or milk. We can also look at the activity that's going on. And so we do this by measuring our potentially mineralizable nitrogen and our carbon mineralization or respiration. And when we look at this, this is providing really important information on our nutrient cycling, biological life, things like that. Potentially mineralizable nitrogen, PMN, is a really great test, but unfortunately, it's pretty labor and time intensive. This test takes 7 to 28 days in order to complete. And so it's not a common test to find in soil health testing packages. There are a few labs out there that do offer it, so you can get it, but you're more often going to see respiration and mineralization as a test that you can get from a lab. And we can think about these tests as measuring the trash. So if there's a party going on at a house, we can look at the trash in the trash can afterwards to kind of get an idea of what was going on there and how much was eaten, how much food was consumed by the people that were there. And so the idea is that the more microbes we have, the more trash that it's produced, and we're going to get this higher PMN or carbon mineralization value. And so for that, we do want to have a higher value. And then we have this last kind of small pool of immediately available carbon and nitrogen that is extracted using water. And this is commonly found in the Haney test. And the goal with this is to mimic the soil water matrix and in the environment that the plants are actually experiencing to get an idea of what these plants are actually able to take up immediately. And so we see a lot more fluctuation in this pool of carbon and nitrogen. It serves more as like kind of a snapshot in time. And we can think about this to continue our analogy as like a bowl of Doritos or chips, where you have to do no prep really to get them. You open the bag, you're good to go. It's pretty much immediately available. And so these pools serve to form as our kind of organic matter and kind of provide us that insight into what's going on with this indicator of our soil health. And so as we kind of think about all of these tests and how they're actually going to be used to tell this story about our soil, we can kind of think about it on this timeline where we have these more stable, less varying tests like organic matter, total carbon, total nitrogen, things like that. And those tests are going to be better for our long-term monitoring, farm planning, and comparing to our benchmarks. On the opposite end of that spectrum, we see these more sensitive tests like the water extractable, organic carbon and nitrogen, which are going to be more sensitive to changes in management. And so we can use them for the early detection and comparing some of our on-farm changes. And that's not to say that one is better or worse than another. I think it's just an important context clue and good information to have to show where they're going to be most beneficial and trying to investigate what's going on in your soil health. The next group I'm going to talk about, and I'm going to talk about this pretty briefly, are our chemistry and nutrient indicators. Um, I'm not going to spend too much time on this, mostly because they are pretty well-established tests, and there's a lot of resources already out there for understanding what these fertility tests are going to tell you. The 2 points I really just want to make, one is that you really need to know your crop when looking at these tests. Different crops, of course, have different nutrient requirements and nutrient needs. And so to know whether or not you have enough potassium is going to depend on what crop you're trying to grow. And so if you're not sure kind of what that might look like, there are a number of great resources out there, one of which are these California fertilization guidelines. You can go in here and it has great information about when to sample, what to be looking for, things like that. The second point I want to make is that these nutrient extractions use extractants that can be pretty regionally specific. And for that for that reason, it's important to, when possible, try and get that tested by a local lab. Your local lab will have techs who are aware of the different soil variability that occurs in your region and can adjust their extractants as needed in order to get the most accurate results. That's not to say that an out-of-state lab won't do that or can't do that, but your local lab will be much more aware of what's going on in your area and can help strengthen your interpretation of those results. Next, I'm going to move on to aggregate stability. Aggregate stability is measuring the physical habitat of the soil. And so we're measuring the soil's ability to resist degradation when exposed to physical forces, such as tillage, water, wind, things like that. And aggregate stability is really important because it contributes to good soil structure. And this includes not just the fact that your soil has pores, but also that your soil can maintain these pores. And so this really cool aggregate diagram here kind of shows what all goes into this in order to make a really great aggregate. For anybody who was here last week and heard Tom Willie speak, he was talking about how this is kind of like a planet. And so we have all of these different parts in this aggregate that lead to this functionality that provides a really important role in our soil. And so in this aggregate here, we can kind of see these smaller microaggregates. These microaggregates are really important for soils, water storage, stability, filtration. It also provides habitat for fauna and roots, microbes, things like that. And all of these microaggregates are being held together by these chemical and physical bonds. And they're holding in here this plant and fungal debris, this organic matter. And that is really important. It's holding this all together because that's going to help resist erosion and things like that. And so these microaggregates that are many millimeters small are also playing a really important role. We also have these red sections here. This is decomposing particulate organic matter, or POM. This organic matter is derived from plants and animals, and it's being broken down by this fungi. You can see all the little lines in here that indicate that. This organic matter is really important for nutrient cycling. It supports microbial life and activity, and it also has important implications actually for our soil chemical health. Um, Soil pH buffering capacity is influenced by particulate organic matter, and so it has multiple different roles beyond just aggregate stability. Then we have all these green lines here. These are the mycorrhizal fungal hyphae. You've probably heard a lot about these. It comes up a lot when we talk about plant and soil health. This fungi is really important for forming a symbiotic relationship with the plant roots to help with nutrient uptake. And we're also seeing the formation of this organic binding agent, the glue that's kind of holding all of this together. And that's kind of this beigey taupe sort of shade that's in the background here. And then we have finally this like little kind of lavender lilac section. This is just empty open pore space. That space is also important because that's where we're seeing the water and the air move through our soil profile. And so in this small, zero point 0.6 millimeter aggregate, we have all of these really important functions going on that are going to play a really important role in our soil. And the formation of these aggregates is affected by a number of different things, some of which are management related, so tillage, adding in organic matter from cover crops or plant biomass will affect it. But there's also a really large influence of our inherent climatic influences. And so those are things like rainfall and temperature, soil texture, all play a role. And part of this is because our aggregate stability is influenced by soil organic carbon. And in regions with cooler temperatures and more rainfall, are going to have a higher rate of soil organic carbon formation versus areas that are hotter and drier. And so we see that places like the Central Valley, California, where you do have those hotter and drier conditions, have a slower rate of SOC formation and therefore have a lower baseline aggregate stability. And so I want to point this out because if you're looking at tests and results from different areas, it's a really important contextual clue to be like, okay, just because somebody in the southeast has this aggregate stability and they have a clay loam soil or a sandy loam soil, and I do too, and we're both doing the same thing, why is it so different? And so you just want to kind of keep that in mind when looking at your results for aggregate stability. And when we measure aggregate stability, there are a number of different ways we're doing so, and they're kind of measuring some different things. We have the rainfall simulator and wet sieving here on the left. And these two tests are measuring the soil aggregate's ability to withstand outside destructive forces. So this is the rainfall that we're measuring its ability to resist. On the right here, we have the Slakes app, and this is measuring slaking, which is a little bit different. And that's focusing on the aggregate's ability to withstand internal stresses. So if you have kind of a rapid uptake of water, the clay in the soil will swell, the trapped air inside those aggregates will try to escape. And we want to measure how well that soil aggregate can resist that. And this is a really important measurement because slaking is what is a primary driver of our surface sealing, which leads to flooding. So when rainfall hits our topsoil, it dislodges those soil particles, which then settle on top and kind of seal up our soil and prevent water from moving through. And so we're measuring just different types of aggregate stability. All of them are important though. Um, Kind of diving into these two tests a little bit more, the rainfall simulator was developed by Cornell. It is, again, one of those tests that's a little bit harder to find out and about in labs just because the apparatus isn't common. There are still a couple of labs that are using it. Wet sieving is more common. The results between these are pretty comparable, so it's not, a huge deal to use one or the other. But again, just kind of make sure you're sticking with the same one. Both are pretty good at distinguishing between different management strategies. So reducing your tillage, increasing your organic amendments, things like that. They've both been shown to be able to distinguish between those. The Slakes app is great because you can do it at home. You can see here it's just kind of using a phone, piece of paper, and some aggregates. I use a soup can when I do this, so you can use pretty accessible materials to carry out this test. You can still get it done in a lab, but it's a really great thing to use when you're at home. What's important to note, though, is that your results are very much impacted by the way that you handle your soil. And so, a lab will handle soil differently. You might handle your soil differently. But what's really important is to know that it's done exactly the same as much as possible every time. So, if you're doing the Slakes app at home, take your sample, let it air dry for a week, and then run your test. Sending it to a lab, just make sure you're sending it to the same lab each time and they're using the same method each time. With all of these different options, there's no one perfect one. They're all pretty good. They all can help distinguish between different management strategies. So it's not like one is particularly worse or better. They do have a little bit of a difference in sensitivity. Wet sieving is looking a little bit more at the long-term structural stability of soil, whereas the Slakes app and Rainfall Simulator are looking more at some of these like early detection and vulnerability to erosion and things like that. So there's a lot of overlap, but again, not hugely so. And then this last section of the test we'll talk about for a little bit is microbial life. We've talked about the food that's available and how active they are when we were talking about these organic matter pools. But I do want to take a little bit just to talk about understanding who's there and what they're doing. So there's three kind of main tests that we use when looking at microbial life. One of them is PLFA. This is our phospholipid fatty acid analysis. Different microbes in our soil have these kind of distinct fatty acid profiles. And by measuring them, we can get an idea of our broader community profiles and the functional groups of that life that's in our soil. Um, One thing to note about this is that we don't really have a benchmark for like what we should be aiming for any of the tests that are in this analysis. We know that more is generally better, but we don't really have a set like, okay, after you hit this point, this is where you're hitting healthy levels. And so what we use this test for is for comparing between management strategies on our farm rather than against a benchmark. The second group is our microbial biomass, carbon and nitrogen. For this test, we're looking at the amount of extractable biomass in our soil. So that's bacteria, fungi, protozoa, etc. And we're looking at this because it indicates some of our soil's ability to cycle carbon and nitrogen. And so again, we can use this then to use as a comparison between different management strategies because we just don't really have a great benchmark for what we want. Again, more will be better, but we don't know at what point you kind of hit that level. And then the new kid on the block, and one you may have heard about frequently in recent years, is this DNA sequencing. And so labs are using kind of high throughput sequencing techniques to assess microbial biomass. And so they're looking at the diversity of the different microbial life that's in our soil, ID down to the genus level. And so this is helping us understand that if we have, 1,000 microbes in our soil, is it 1,000 of one or two, or is it 1,000 of eight different species? And so we're trying to kind of understand how many different species are there. Kind of the problem with this is that because we're only IDing to the genus level, it's not giving us a ton of like really good in-depth information about what exactly the roles of those microbes are. So it's like someone comes to your farm, tests your soil, and tells you you have Fusarium wilt, but like doesn't give you any more information about what type of Fusarium wilt you have. You don't know if it affects your strawberries, your tomatoes, another crop, things like that. And so we're kind of left with like some questions about, okay, now what? And like, what does this actually tell us? Some companies are using it for prediction of risk, for yield improvement, things like that. But there's still some question marks about what the role of this test is in our soil health testing. And they all kind of fluctuate, fairly quickly. There is some kind of movement through here depending on what aspect of these tests you're looking at. But we do generally see fairly quick responses in our community makeup as a result of changes in management practices within these tests. So they can be good for detection of changes, your on-farm comparisons, monitoring, and things like that. Okay. In the interest of time. Okay. Oh, great. We're doing okay. The last kind of section I want to talk about now is just how do you do all this? Like we've talked about all these tests and thinking about how we can actually use them. I mentioned a couple times that we don't have regional benchmarks for what soil health looks like. And so it kind of leaves us in the lurch of like, okay, like great. Like if I'm doing all these things, how do I know how well I'm doing? Or how do I know what effect all my work is having? And so if we don't have these benchmarks, what we can do is create our own internal benchmarks. And we can do this on our own farm. And there are three main steps that we can use to investigate that. The first is to determine the question. So what do you want to know? Do you want to know the effects of cover cropping? Do you want to know what soil health practices can help, can soil health practices help with flooding? Or are my soil health practices just like having an effect in general? And so identify that strong question. And so for the sake of today, let's say I want to know what is the effect of cover cropping on my soil health. Now I need to identify a comparison. And so I take a field that I have been cover cropping for 5 years and I compare it to a field that has not been cover cropped. And I generally want to make sure that the soil texture is the same and that, some of these other things are held similar. But then I can take those two locations, test the soil, and then see what results come about after that. And hopefully that'll give me an idea where I can compare my cover cropped field to my baseline field and see what that difference is. And an important aspect of this is trying to make sure that when you do that you do that sampling at the same time of year. It's not necessarily a matter of I want to sample every March 3rd. It's more of I want to sample when it's, between 50 and 60 degrees outside. I had rain a week ago and I haven't terminated my cover crop. This is going to help you sort of maintain some of these conditions that would affect your results. And so try to make sure that you're sampling every single time during those same conditions. Try and get an idea of what's going on. And so if we look at this within the context of a case study, we worked with a diversified vegetable farm in our area who had been experimenting with leaving fields fallow for 3 years and then integrating chickens into that as a rotation. And their question was, is my fallow and chicken rotation with annual vegetables having an impact on my soil health? They wanted to know if it was kind of worth that labor. And so we set up a comparison for them where we took that field that had had the chicken and fallow rotation and compared it to an adjoining field that had not had that same rotation, but was otherwise the same. And so we took that soil and submitted it to a lab. And what we found was that the field that had had the pasture on it, these blue bars with the circles around them, were higher in these different indicators of soil health. And so when we compared it to their other field, that baseline field, they were able to say, okay, I can see that my practices are actually having a positive impact, and I'm seeing an increase in these soil health indicators. And they could decide that, yes, the work they had been doing was worth it, and that all of that work that they had done was validated by this data. And so they used their own internal system in order to see how much of an impact their practices were having, and they could benchmark within themselves. And so, in this way, we can use soil health testing in the absence of benchmarks to answer our questions and explore some of these soil health questions that we're grappling with and kind of figure out how we can use these soil health tests to work for us. And to kind of help support that, I'm going to put a quick plug in here for some tools that we're developing. We're trying to develop a infield soil health assessment so you can test your own soil health at home. We're also trying to develop some soil health report app availability so that way we can collect soil samples and help create these benchmarks. And then we also are developing a soil health selector tool. And the goal of that is that you could ask this question of like, I want to know what tests will help me, you know, understand the effect of cover cropping. And so you could go online and you can answer this set of questions, and it'll help guide you through that process of figuring out what tests are going to work best for you. So there's a QR code here, and we'll drop the link in the chat. But if you're interested in helping us test these tools, we would love to have assistance. If you're just interested in getting the tools once we're done, we would also love to have you to partake in that aspect of it. So, yeah, please help us with all of that. And we're excited to share these resources coming up. But with that, I will take a break and we can do a Q&A because it was a lot of information pretty quickly. But thank you, everybody. And yeah, we'll go from there.
Thank you, Lindsay.